I'm going to give you an update on what's happening on the forefront of genomics and show you the quick history of time as things are changing to see how fast they're changing. But just let me mention this is a rendering of my new institute building on the UC San Diego campus. It's being built. It's going to be the first zero carbon research building in the world. So we're trying to actually put some of our new ideas into actions, and including the experimental nature of the building itself. Now, what we've been doing for several decades now, when we read the genetic code, we go from this 4-letter analog molecule into the ones and zeros of the digital world. And I call this digitizing biology. And this is happening, as you know, at an ever-increasing pace, starting, as you heard, in 1995 with the first complete genome of a living organism, followed only 5 years later by the first versions of the human genome. And then things have obviously scaled up dramatically from that point on. This is the team at the White House in 2000 with the announcement with President Clinton and Tony Blair. The person on your left is Mark Adams. The tall gentleman is Ham Smith, who got the Nobel Prize in 1978 for discovering restriction enzymes. Next to him is Gene Myers, who led the team to develop the new algorithms. And these are the scale labs that we had. We had 300 of these $300,000+ machines to sequence the first draft of the human genome in 9 months for about $100 million. Technology has been changing rapidly, both in computing and in sequencing. In 1999, we had to build the world's third largest computer just to handle this new genome data. It was only now 1.5 teraflops with 100 terabytes of storage. All this is changing. We just last year celebrated the 10th anniversary of the human genome. And this slide just proves that people look better in tuxedos. Now, the technology has changed even faster than we've aged. Those giant rooms now have been reduced to single boxes, and those boxes are changing. This is the recent announcement from Life Technology of the new ion proton sequencer. And this kind of technology is truly an amazing development. The chip in the lower left is the actual sequencing machine. It's basically a pH meter layered on top of a computer chip. And what happens, as I'm sure you know, is when you add a nucleotide to a growing chain of DNA, it releases a proton. So all that machine does is measure that single proton release in each of those tiny wells and registers that. So it's technology getting simpler and simpler, faster and faster, and cheaper. And this machine supposedly can sequence an entire human genome in about 2 hours, which starts to change how sequencing can be done clinically. And instead of a $5 billion public program or a $100 million private program, it's now on the order of $1,000 or $2,000 to sequence a human genome. It probably costs $10,000 in compute time to analyze it currently, but that's changing too. That giant room of computers we had, now you can get a single board to add to your laptop or desktop computer. Each one of these can do over a teraflop of calculations. It becomes important these new machines generate so much data, even though we have dedicated optical fiber between the two campuses of my institute in La Jolla and Rockville, Maryland. It is now faster and cheaper to send data Federal Express than it is to send it over optical fibers, just because there's simply so much data and information. Genomics took another leap in 2007 when we sequenced the full diploid genome. What was done in 2000 was only half of the genome. And doing the full genome, we found that human variation was much greater than people thought, going from 1 in 1,000 base pairs to 1 to 3 per 100 base pairs. So substantial difference as we start to analyze human variation. And for the first time, we're able to start to do haplotype phasing. This is going to be very important to the future of work as it matches up with clinical tests. And what haplotype phasing is means we can separate the two sets of chromosomes, the one you get from your mother and the one you get from your father, in the computer, versus just having it just be a collection of bases and genetic changes. Because understanding what you inherited from each parent and what you pass on to your children is a very important part of understanding the impact of genetics going forward. The technology is changing so fast that we can actually sort single cells. This is actually sorting sperm cells. We can do a human genome on a single sperm cell, which is one way that we do haplotype phasing, sperm being just a haploid genome. Vanessa Hayes at the Venter Institute has also been working on sampling global diversity, working some of the earliest populations in Africa. She's headed the team that sequenced the Bushman genome and Desmond Tutu's genome, and comparing the Bushman's genome to my genome to a Han Chinese from the Beijing Genome Institute. It comes all within this range of the 1% to 3% variation. However, just looking in Africa and the different groups there, something that's been known for a long time but never really been quantitated, there's actually much more genetic variation within Africa than between Africans and any other group. Getting back to the nature of being the origin of humanity, some groups migrated out of Africa, then migrated back, so there's been a lot of change. All this is adding up to beginning to understand the correlation between genetic changes and traits. In this case, in this slide is just showing a very recent trend, even though the pharmaceutical industry has sort of resisted using genetics for clinical trials. Some recent things such as Pfizer's lung cancer drug, finding that the clinical trial failed until they went back and looked at individuals with mutations in the ALK gene, a tyrosine kinase receptor gene, and found that this drug worked on over 60% of those patients in reducing tumor size. There's obviously increasing numbers of treatments associated with rare genetic variants, but this is just the earliest stages of this whole area. Now, in addition to your 100 trillion human cells, each of you have about 200 trillion bacteria. Even if you did shower this morning, you still have them. And when we first measured these in 2003, using the same techniques we use for sequencing the human genome, this became known as the human microbiome. So we have around 22 or 23,000 human genes, but if you look at the genes associated with all your microbes, you have about 10 million additional genes associated with each one of us. Just a variation of thousands of different species in your mouth and different body cavities in the skin, etc. So a big question has been to develop what effect do all these genes and all these microbes play on human health? And there's now widespread studies looking at the microbiome in human disease, finding an association with the diseases listed on this slide, in addition to multiple others. When you look at changes in cancer in the US since 1975, one cancer really stands out, and that's esophageal cancer. When we look at the microbiome in esophageal cancer patients versus normal ones, the cancer patients have a totally unique microbiome in their esophagus. It's not 100% clear yet whether these microbes are the cause of the cancer or are resulting from the change in environment, but these studies are ongoing and just typical of the early hints of things. This is a study that Karen Nelson, who heads this team at the Venter Institute, did with UCSD, showing that In fact, the toxic effects that alcohol has on the liver, apparently some of you experienced that from your party last night, is not due to alcohol directly affecting the liver, but alcohol affecting bacteria which can release toxins that can damage the liver. So not being a physician, I've come up with a simple prescription. I think we should just fortify alcohol with antibiotics, and then you won't have any liver disease. But nobody's picked that up yet. So looking at humans, we have to combine all this information. We have to look at the microbiome, how your genetics affects which bacteria you have, how the bacteria affect the expression of your genetics, etc. Some of this comes together also looking at stem cell genomics. Many of you are aware that you can now make stem cells just from skin biopsies. These are called iPS cells, and these iPS cells can differentiate into a wide variety of human cell types. So because my genome had been sequenced, they wanted to look at my stem cells from this process early on. So from a skin biopsy, Goldstein's lab made iPS cells, and the standard in the field was shown in the bottom of the slide. They looked at the karyotype to see if stem cells had a normal karyotype. That's like trying to work out what country you're from as an indication of your health. These iPS cells clearly had markers for all three germ layers. Larry's team generated neuronal stem cells from these skin cells and then turned these to differentiated neurons. So there's groups in San Diego looking at my brain cells in tissue culture. And the point of trying to do this is using stem cells as a means of studying diagnostic procedures and medical treatments, drug treatments on cells instead of doing this. I wasn't willing to give up a brain biopsy at this stage. This was much less invasive procedure. But by looking at my neurons in culture, comparing it to my genome, certain predictions perhaps can be made, and also testing different drugs on these neurons. You can also differentiate stem cells into making liver cells. So perhaps a key part of future clinical trials will be testing drugs on a patient's stem cell-derived liver cells in culture versus doing the initial studies on patients. The problem with this area, though, right now, as we found from comparing the genome of the stem cells to my sequenced genome, is that cells during this iPS process and in the culturing process develop multiple mutations. And this is starting to show up now as a general theme. And here's a group that looked at 125 embryonic stem cell lines and found the longer they were in culture, the more mutations they developed. So it's not just a problem with iPS cells. And so the question is, where do these mutations develop? Why do they develop? What's unique about cells in the environment of our body that maintains the genome in a steady state versus when you change the environment, so methylation changes, which can lead to new oncogenes, et cetera. And so the question is, can we understand these changes and prevent them? And then if we can't prevent them, can we repair them? And so this is where some of our work that I'll mention in a minute on synthetic DNA comes in. How can we actually make genetic corrections, basically doing gene therapy on isolated stem cell lines before reinserting them? And some groups have had success with this. And here's a group that's looking at stem cells derived from a Parkinson's patient that had genetically inherited Parkinson's disease. And it's also a nice way where you can use a patient's own cells as the control and the experiment. So they took parallel lines and they changed the genetic code in the gene that causes Parkinson's disease in these cells. And we're able to then use the other cells as a control and start doing numerous studies. And here's another one that even though cells undergo lots of mutation in culture, they found that by correcting some of the mutations, at least those processes didn't cause additional mutations. So as a patient advocate, I'm not ready to have these neurons put back in my brain, even though as I age, I would like a few extra ones. We need to find a way to solve all these problems, and it's even important if you're going to start testing stem cell-derived liver cells or neurons in culture, you want to make sure they in fact represent the patient, and not just a mutated form that don't really represent the individuals. Every iPS cell line obviously will need to be evaluated, but also embryonic stem cells look like they will too. So I mentioned that we can do single-cell genomics. We now have Roger Laskin, who's developed this technology, has developed a high-throughput platform for doing genomes on single stem cells, single human cells. In fact, we can actually do single-cell transcriptomics, looking at gene variations and gene changes in single stem cells, as an example. So how do we get to this next stage? There's lots of groups now scaling up to sequence more and more human genomes as the cost goes from millions down to thousands, perhaps down to hundreds, where you can get your whole genome done for faster and cheaper than you can get a BRCA1 gene test. Things are going to change, but the main problem we have is interpreting this data. The number of genetic studies going on of any meaningful nature are extremely limited. So the only approach that I think is going to work out is to really scale this up. We obviously can do thousands of patients for substantially less than the original genome sequencing, and we need at least 10,000 individuals to have their genome sequenced. But that sequence on its own is meaningless unless we also get the complete human phenotype, and that's yet to be described. So that's one of the biggest challenges going forward, everything from drug responses to disease characteristics, things that's even hard for the clinical world to decide on universally. And all this information has to be digitized so it could be compared in large computer searches back to the genome. One of the other exciting areas we're working on that you heard is synthetic life. We started looking at this simple genome with about 500 genes, trying to work out how many are essential. Ultimately decided we had to design and construct a genome to be able to answer these questions. And then we had new questions. Would chemistry even allow making these large chromosomes? And if the chemistry allowed it, could we even boot them up? Our first study was published in 2003, where we made Phi-X174. We developed new techniques for correcting the errors in DNA sequence. Starting with the digital code in the computer, we made the 5,000 base pair genome. And the next stage is we inserted that into E. coli. E. coli recognized the synthetic piece of DNA, started making proteins. The proteins self-assembled and made the virus. The virus showed its gratitude by killing the cells, which is how we detect them with these clear plaques on a plate. We call this a situation where the software is building its own hardware, because all we did was put in the software and it made the virus. We didn't want to make viruses, we wanted to make whole self-replicating cells, but we thought if we could make viral-sized pieces accurately, we could find a way to put them together. It took us several years to do this, but we found a way by, in the lab, assembling these pieces, cloning them, sequencing, and going on to the next stage, we could make linear progress. We found then by putting large pieces into yeast, that yeast would automatically assemble these pieces together using homologous recombination. And a few years back, we reported the first complete synthesis of a bacterial chromosome. This was the largest molecule ever made of a defined structure at the time. We continued to improve the synthesis process. Dan Gibson came up with his invention of being able to just take small fragments of DNA designed carefully, put into a single tube with 3 enzymes at 50 degrees, and it assembles them automatically. And this allows us now to robotize this entire process, going from the digital world to the chemical chromosome without much human intervention. And this was initially tested and proven by making the entire mouse mitochondrial genome. And mitochondrial genome therapy may be a new avenue going forward. So how do we boot up a chromosome? The study we published in 2007 we think is one of the most important because it showed simply by changing the DNA software in the cell, we could convert one species into another. So we did this by isolating a chromosome from one bacterial species, Mycoplasma mycoides. We removed all the proteins from this DNA. We added a couple of cassettes to it so we could select for it, and it would turn cells bright blue if it was activated. And then we developed new ways for injecting that DNA into recipient cells, in this case, M. capricolum cells, which are about the same distance from mycoides that we are from mice. We have this very sophisticated movie to show you what happened. We injected the mycoides chromosome into the capricolum cell. Now we have a cell body with two sets of genetic instructions. And what we think happened is the DNA that we put in started immediately being read. It produced numerous proteins, including restriction enzymes, that recognized the original chromosome in the cell as foreign DNA and chewed it up. So now we have the body of one cell and the software of another. In a very short time, we had these bright blue cells. And when we interrogated them, they only had proteins coded for by the chromosome we inserted in the cell. Any trace of the original species was completely gone. Everything was driven by the software in the cell. So if you're not convinced yet, life is a DNA software system, and if you change the DNA software, you change the species. We took this further by being able to clone entire bacterial genomes in the eukaryote yeast, simply by adding a yeast artificial centromere to the bacterial genomes. And this allowed us then to make rapid changes in bacteria that you couldn't make otherwise using yeast genetic tools. We perfected ways to get the DNA out of yeast and into bacteria, and learned that the DNA had to be methylated. And we were finally ready then to go at a full blast at getting a synthetic cell. We decided that with these new techniques to make the much larger M. mycoides genome. It's 1.1 million letters of genetic code. We synthesized that in a matter of weeks, injected it into the cell, and it didn't work. And we went back over a period of time developing new tools for finding errors, and we found a single base pair deletion that made the difference between life and no life. When we corrected that deletion, we got the first living self-replicating cells driven entirely by a synthetic chromosome designed in the computer and made with 4 bottles of chemicals. So this was reported in 2010 and represents the first synthetic life, which helps us understand the genetic code and its relationship to biology. Very quickly, we're using some of these tools in the clinical area, particularly in vaccine development. We formed a new company called Synthetic Genomic Vaccines, and we're working with BARDA and the US government and with Novartis. Building on the experience we've had with Novartis, we developed the type B meningitis vaccine that's just coming out in Europe this year. We now very rapidly make synthetic flu viruses for vaccine development. You might remember with H1N1, it took about 9 months to get a vaccine out. We wanted to reduce this greatly, and perfecting these techniques of DNA synthesis. BARDA now sends us a test sequence of a potential pandemic flu virus, and within less than 24 hours, we synthesize the new variants of that, send that to scientists at Novartis who get it ready for expression and production in their NBK cell facility in North Carolina. This entire process we now have down to about 5 days, but that includes transportation time, so we think that will get better. That's a fast tour through genomics. We're able to write the genetic code now for the first time, and I think the implications of this for science and medicine are just beginning to be felt. Thank you very much.